Welcome back to Biomandscape. Today we are going to know about sporulation. What is sporulation? When there is a harsh environment or too dry or too cold or too hot environment, the parental organism they divide internally into many small offspring through multiple fission. So each tiny new offspring it have a layer, a protective outer layer that is resistance coat or a cyst. We call it resistance coat or a cyst. So it is a tough layer which is present for protection. So the entire group of the coat they are coating the offspring and now they are known as the spores. Now what is the dormancy? The parental structure they break open releasing those spore and they remain inactive and almost like they are in the hibernation until the condition is improved or favorable. So when favorable condition occurs, when the environment becomes suitable again, like warmer or more moisture is available, the protective coat it breaks, opens up, and the spore grows into a new adult organism. So sporulation allow organism to pause the life cycle and wait out in the bad condition, ensuring their survival and helping them spread to the new places where the time is right. Now, what is uniparental? A condition when a person receives two copies of chromosome only from a single parent and not a copy from the other parent, so that is uniparent. Now fragmentation is a form of asexual reproduction where an organism is split into a fragment, and this fragment they develop into a mature fully grown individual which follow the mitosis, and it occurs in spirogyra, fungi, and some annelids and sea star. Now, regeneration, what is it? It is a process of renewal, restoring, and regrowth. Growth. It can occur at the level of a cell, tissue, organ. So, it is common in Hydra, Planaria, flatworm, and echinodermids. And lizard can discard a part of its tail when it is in danger. So, the tail can regenerate later. So, in human also, the liver it can regenerate if it is partly damaged. Going to know about polygenetic inheritance. It was suggested by Galton in 1883 that many instances of continuous variation, these variation they are heritable. So he thought that tall human being they produce tall children. They do not produce the shorter one. And he suggested that this character like height, mental capabilities in human, they are heritable, although they show a continuous range of variation in a population. So Galton's postulate it gained experimental support when it was found that at least in some instant some character they can be determined by more than one gene. So each of which the same but cumulative phenotypic effect can be seen, like quantitative character like height, the plant height, the yielding of the crops, the size of the crop or the shape and the number of the seeds and the fruits per plant, like intelligence in human being and milk yielding animals. They have been found to determine by many genes, and their effect they have found to be cumulative. So each gene have certain amount of effect. So more is the number of dominant gene, the greater will be the expression of that particular character. The quantitative inheritance that is also known as the polygenetic inheritance or multiple factor inheritance. Though polygenetic trait can easily influenced by the environment, these are controlled by three or more genes with phenotype reflecting the contribution of each allele, like quantitative. So polygenetic trait it can be studied by inheritance like in human skin color. There are no contrasting phenotype for the trait. Let us assume that this trait is controlled by three genes like X, Y, and Z. So in this cross, there is a mating between the dark skin and the fair skin human being. And then the intermediate skin color individual that is obtained from the cross is F1, and the F1 it is mated to obtain the F2 progeny. So we can see in this diagram where there is a P generation, parental generation, the cross is made, F1 generation is seen, and F2 generation we get it in the ratio 1:6 15 20 15 6 1. So it will show a bell-shaped curve. Few individuals fall into the parental category. We can see the expression of level of phenotype is dependent upon the number of contributive allele, and hence it is more quantitative. So there is a cross that depicting the inheritance of human skin color, they are controlled by the polygene. So in the relative frequency of two progeny of the polygenetic cross, it is plotted against the extended of phenotypic expression. A typical inverted bell-shaped curve is obtained. So it is clear that the expression level of the phenotype, it is depend upon the number of contributive allele, and it is more quantitative. Few individuals fall into the parental category. So in the F2 data, it is plotted graphically and a bell-shaped curve is a result. So in this example, we have assumed that involvement of the gene pair. It can be higher number of genes they are involved in determining the phenotype, and greater variety that might be expected in the F2 generation. Other example that can be studied like kernel color in the wheat and inheritance of the comb length in the maize. So it is generally believed that during evolution there was duplication of chromosome or the part of the chromosome that result in the multiple copies of the same gene. So in large number of character, they are controlled by the polygene in which the allele contribute adaptively to the phenotype. So this result in the polygenetic inheritance. So for knowing such concepts, follow Biomindscape. Thanks for watching. Do like, share and subscribe.